It's a nematodes world. Actually, the title that came to my mind because it has been said by Nathan Cobb that if you remove everything from Earth except the nematodes, then also the world will be dimly recognizable because they are ubiquitous, found everywhere. Even the mines which were actually, they went up to 1 kilometer or further deep and they could find nematodes, but no other metazoa except bacteria and other microbes. But so that's why they are very hardy and everywhere they are found, and that's why their importance is immense. If you see on both sides, whether they are friends or foes, because they are parasites, plant parasites, animal parasites, found in all types of plant species and animal species. Then on the other hand, if you see, they also are killing the insect pests, because being parasitic, they are killing them. So they can be used in biocontrol programs, and of course, they are killing many harmful bacteria, being the bacteriivores. So whether to assess whether they are good or bad, it's up to us, because how can we exploit them? So for all those who are not from this, actually, who are not very familiar with the nematodes, it's a very, you can say, it's a worm which is showing high diversity, microscopic to very large, about 8 meters long. That is the parasite of whale. So that much is the size diversity. And they have been named as threadworm, eelworm. You all must be knowing about the C. elegans, that is considered to be the excellent model. So this is one nematode that everybody knows. But how these nematodes behave, how are they in the environment, what are their allies, they are all, that is very, you can say that they are coming very close to arthropods because they are ecdysozoa. They are showing molting. So here I would tell you some of the basics also, because while doing the study on nematode, I'm just working on their taxonomy. So we are to know the species in its intricate details. So if you see here, that is, they are occupying 50% of them are marine, which have not yet been explored. Very hardcore. You can find them everywhere, even the hot water spring, the volcanic vents, they have reported the nematodes because they were collected from there. And you can see the C. elegans, which was taken to the space for some research experiments. When the space shuttle Columbia crashed, then also they were live. They could be traced. So this is how much hardy they are. But they constitute a component of the biodiversity, and we are mostly, when we talk about biodiversity, we are mostly concerned about the above ground diversity and the below ground diversity, which is basically a huge, you can say, domain that is usually unexplored or that is usually ignored. So this is a lopsided approach, because mostly we are not working on the below ground diversity, which is basically the source of sustenance of all of us. So the exploitation and soil degradation, they are actually very critical threats for this below ground biodiversity. Now with nematodes, if you see, they are in environment, they are actually very helpful in bioindication. That means they are showing different types of feeding gills, which is very rarely found in any group, because they can be herbivores, they can be fungivores, they can be bacterivores, they can be omnivores as well, and predators. So if you do the community analysis, this is actually the cheapest test that you can conduct for any environment to detect the quality of environment, because the tests which you can conduct here, you can see them in the food web. That means at every stage, you can say at the level of predators also, here you will see that they are herbivores, they are also the first degree of consumers and second degree of consumers. So you can see at all levels they are present, and that is basically very good indicator. And we just rate them through various CP scales, that is colonizer persister scale, that is whether they are the ones which are enrichment lovers, in enrichment indicators, or they are the ones which are the persister, R and K selective, you would have known. And so they are in different trophic groups also. So when we do the analysis, if you see the prevalence of herbivores, fungivores, and bacterivores in very high quantity, then you can say that the food web, that is basically resource-rich food web. But again, the bacterivores, if they are dominating, that is actually the decomposition channel, that is food decomposition channel, that is basically bacterivore dominating, or the fungal dominating pathway, they are working. So that means, because when you are taking the sample from desert, you will find that because of the dry substrate, you may find that there the bacterial decomposing pathways are working. Likewise, in humid climates, you will find the reverse is there, and the community that basically very aptly indicates all these changes. Even you will see that the ratio of opportunists to predators or omnivores, that also will be indicating about the type of food web, whether it is regulatory or it is suppressive type. So that's why they are now very largely used as the bioindicators. So for that, we also conducted a study just to see that how these nematodes are there in different types of contaminated or perturbed type of environment comparing to the natural one. And you can see that the diversity, that is the first thing to be impacted due to contamination or enrichment. And here also you can find that the natural habitats, they actually showed a different grouping as compared to the ones which are enriched. The different types of samples from totally enriched, that means organically enriched habitat, or those samples from food industry, metal industry, paper and sugar mills, they were all collected just to see the impact. And even we could find that the taxa also, they were also showing the difference, because most of the omnivores or the case selective ones, they could be reported in the natural habitat. So we could see that the bacterivores, they constituted in great percentage, and of course the generic diversity that was found to be least in the paper industry samples. That actually could be because of the various type of dyes or chemicals which were more, you can say, because we still are interpreting the results. And here you will find that the MI, which is the maturity index, and the MI 225, they were considered to be very low in the disturbed sites. Some of the nematode species, they could be identified which were anoxic, which were actually very prevalent in anoxic habitats. That also indicated that the contaminated aquatic habitats, they were actually having very, you can say, low degree of oxygen. And again, you will find that most of the enriched habitats, they also showed the nematodes which were associated with insects. That led our study a new direction. So for all these works, the first thing, that is the foremost, that we have to identify correctly the species. The correct identification, that is a must. And this identification, this is crucial, but how many people are working on taxonomy? How many people are working on the identification and the biodiversity studies? That is again a thing to ponder upon. So unless we know the biodiversity, because the biodiversity, if that is not attended properly, this is actually the message to convey, then there are consequences which we are going to face. Even COVID-19, that also showed us some of the lessons. When we were all within the houses, we were just during the lockdown period, you found that the nature that actually bloomed. So this is again another thing that we have to look into. So how to conserve, how to protect any species when we are ignorant about their presence? This is also a question, because when we see, there was a paper in PLOS biology which actually indicated, basically it is a very tough thing to estimate the total species documented. But we estimate that there are about 2 million species documented. But most of the time you will find that taxonomic papers, they are only referring a name with some characters. So a name is simply a label. We don't know much about it. And that's why you can say that our ignorance is just enormous, because we are simply documenting it without much knowing about the species. You know, in 1900, C. elegans was first described by Moppus, 1900. After 74 years, in 1974, Brenner was the first one to work on it. And then later it was introduced as a good model. So this is how we are actually not much knowing about the biology, about its interaction with the environment and other things. That's why whenever we have to plan any type of study related to ecosystem functioning, so we have to be very well versed with identification and taxonomy of it. So when coming back to nematodes, you find they are extremely diverse. You can see all these are the different types of nematodes. Simply they are called as worms, vermiform, but you can see most of them, like the rounded ones, that is actually another one which is plant parasite. So you can see the variations which are in the cuticle. You will find the modifications in the form of bristles, in the form of striae, markings. They are lip region variations, immense diversity, and that actually is very much, you can say, amazing in these group of nematodes, and that is showing their adaptive abilities in different types of environment, because they are having their morphology, or you can say their phenotype, very much malleable, very much changeable. So most of the time, the morphological identification, they were based only on the prominent features like lip region, stoma, pharynx, or even the type of gametes or gonads, all these. But many times you will find that some of the cryptic individuals, the ones which are showing continuous variations, they were difficult to differentiate. And that's why these type of problems, or you can see that biology as well as ecology, that was not considered. And that's why a holistic approach, that is the need of time. So what I do, that is because I introduced my work to you, that is we are always working on all the aspects, like just talking about the morphology, the light microscopy as well as scanning electron microscopy, to elucidate the details, intricate details of the nematode. And then one important thing, or one interesting thing, is that the nematodes are transparent. The soil inhabiting nematodes, they are transparent. So whatever you see, that is see-through thing. And that is really the advantageous thing that makes C. elegans as a good model, because even the eggs can be see-through. So you can find that whatever you record, that is, everything is visible. So here you can see differentiation of two species, that can be based on the egg types. They are very close, very, you can say, if you see here, the lip region and other things, they are close. For a layperson, they look alike. But of course, you can say biology offers here the best solution. Of course, we could do the molecular characterization also, which also separated them. But we also went for other type of details just to see their distribution, their association with any insects, or their interaction with other species in the samples. Likewise, you can say another one. What we come across, this is really of great concern, that in the GenBank, many of the sequences which are deposited, they are deposited on wrongly identified species. This is actually a matter of concern. Whenever you compare and you find that, because many of them are para-taxonomists, they are simply the technician or technical people who are not having a proper idea about the morphological characters which may be forming the high weight characters for species identification. And here is the problem, where the sequences which are based on wrongly identified species, they are being compared with the ones which are the genuine ones. So this is actually a problem that we are coming across. So when we talk about diversity, you find that this diversification throughout animal life, that is not distributed equally, because here you will find that the nematodes, they are showing this diversity, which is very astounding. I focus mostly on the group Rhabditida, because it's a vast group and you will find they are different. So here also you can find that the individuals can be having different types of stoma, the feeding apparatus. The ones which are plant parasites, the ones which are feeding on the bacteria, the ones again the bacteriovores or the predators. You can see the variations, and these are the plant parasites. Within them you can find these variations are there. So this is actually, there is a lot to offer if we are only focusing on their diversity. So besides that, I also tried to work on the related areas just to get more information. Like, if the bacteriovores I am working upon, so is there any type of food preference for these bacteriovores? Because that led to a new idea of microbiome. That means the nematodes which are having their microbiome, if we can use that microbiome for identification of the species. That was actually an idea that after, after performing this study, because here we could find that nematodes do find their favorite bacteria, and they are just feeding on them. So there were different species of bacteria which could be collected and which could be identified from the nematodes, and some of them, they actually were very much these were actually the number of colonies, but there was E. coli which was there, and of course you will find that there were two other bacteria, Serratia. Serratia was considered to be using the Trojan horse mechanism. That is, it was releasing the VOCs which attracted the nematodes. The nematodes went, attracted towards the colony, and then died because of the toxic. So you will find that they are also such type of, even there was one experiment which could not be completed, that was still where we could find that the pathogenic fungi and bacteria, they are having some sort of collaboration, you can say, in a way that the bacteria, they are indicating the fungi to form the traps for nematodes. So that was a tritrophic type of interaction that we could find. Still, we are just looking forward to complete that work. So besides that, because these are the supplementary things which I wanted to club with my taxonomic study. So we could also find the behavior of the nematodes, that how the nematodes are performing, and the diversification, if there is some sort of evolutionary relationship between those, because when they are sharing the same type of habitat, so they have to diversify their niche. They have to change their morphology or phenotype also. And let's see, like here you can see this is a group of nematodes which are again coming under the Rhabditida, but they are showing a lot of variation in their labial structure, which are called as proboli. So they are showing very different type of outgrowths. The basic structure you can say, but you can see that gradually you will find that these are actually called as labial proboli. These are the main lips, and these are the processes which are arising from the lips. So we try to work on these nematodes. The different stages, the different juvenile stages, right from, because the nematodes, I should tell you here that there are 4 juvenile stages which actually molt to the adult. So in each stage you find that the shape of the labial proboli that kept on changing. So finally to the adult. So this change was considered, and then all the species in the group, they were actually studied just to see the most primitive one, and gradually how the proboli, they became more and more advanced. So again, with the paucity of time, I am not going to explain it in detail, but actually we correlated this type of trait of the change in the probola type, what are the different structures which are associated with the molecular characterization, and we could find that our results, they actually were quite congruent, because they actually showed that this was the evolution of these proboli at different stages. So earlier, if you see, these are present in the desert region. The bacteria or the food crisis may be there may be food stress. So they are actually just to sway the bacteria towards the oral aperture, and this is how they are using these structures. Then we also could find that the nematodes are associated with insects also. That was another important thing, that here we can explore a very different type of relationships. हमारा टाइम तो नहीं खत्म हो रहा है? So this is also a remarkable feature in case of nematodes, where you see that this is actually the embryogenesis, the first, that is the juvenile that hatches out, and then J2, J3, J4, and then the adult. But if at this stage, at the second stage juvenile, if the conditions are not conducive, if the nutrient supply is very, very scarce, if you can find that the temperature goes very high, then there is a tendency towards forming a DOR stage, that is the resistant stage. This stage basically is the disseminative stage, which will look for some biovehicle to get transported from that habitat to another. This is shown by other animals also, but in nematodes you will find, even if you see some of the animal parasites like hookworm, you would have seen they are showing nictation behavior. They can get attached to the any persons who is just barefoot, and they can just pass into the through the blood vessels into the main intestinal site. So here also you will find that this DOR stage was recovered from many of the insects which we actually sampled in the enriched areas, because the insects are there and we could find the nematodes associated with them. And now we are also trying to work out what type of relationship, if it is a temporary type of association, or if this association is a long-lasting association, because you will find that Phoraceae is the simple type where they get transported from one place to another. It can be ectophoraceae, just attached on the external surface, or endophoraceae, where they can move inside. But you will find another phenomenon is necromany, when they wait for the individual to die and then they will feed on it. And then again this necromany, that has led to two important events, parasitism or entomopathogeny. Entomopathogeny is being used in the agriculture because they are transporting the bacteria, which are pathogenic bacteria, into the insects, and these bacteria, they cause septicemia in the insects and kill, and that's why they are very important in pest control. So what we did, we actually tried to review the past literature, that where the nematodes were collected from, was there any association with the insects, and it was really a very interesting type of review that we could find. And besides that, we could also notice some of the nematodes which were actually associated with the insects to have a spatial type of covering, or the plates, you can say, they are present on the nematodes, and they could be you can say whenever the abrasive surface is there, so they can be removed and the nematode can get into that. So they are just using this type of armor, or you can say detachable type of thing. They could also be noticed. That is because they were very prevalent to the habitat where insects were present. We also could find some of the insect families in different group of nematodes. They actually were very much, I'm sorry. Yeah. I mean, 48 species of nematodes could be recovered from this curculinary family of insect. And likewise, here also you will find that the same family that yielded 64 species of nematodes. That is, you will find that in some of the cases, this relationship is almost obligate. You may find it. And this is interesting because what is the basis of that relationship? That again is the next thing to work upon. So even we could notice that the different body parts which were actually invaded by the nematode, they were also there, but on the large scale, the intestine was found to be the one which was more harbored, and then after that, the elytra, that is actually the wing of the insect, that was also inhabited. Variation in the stoma, you can see so many different types of variants which you can find. Yeah. So within the group, when we were exploring the predatory nematodes, because there were so many predators which could also be found in the samples, and we found that the variation in the stoma was immense. But at that point, we could find that there were two types of predators. One which were having long tubular stoma, that is an indication that they are feeding on the microbes or bacteria, while others which are having massive teeth. So those which were feeding on the bacteria, they actually were more dominant in those sites where you will find the enrichment was there. But when the population increased, then these group of nematodes, they dominated. So the two types of stoma, stenostomatous and eurystomatous, they could be identified in different nematode groups. But then we also found, it's not moving. Why it's not moving? इसके सेल डाउन हो गया क्या इसका? I'm just pressing this. वो भी हो गया, हैंग कर गया है, क्या हो गया? Okay. So within the same nematode, there were two types of individuals which could be recorded. That is, we could find this polyphenism in the nematode stoma, that some of they were having stenostomous condition, the narrow stoma, while others were having very wide stoma. That means the two types which actually showed again that whenever the bacteria are in plenty, this type of morph is promoted. Whenever there are other nematode populations are there, then this type of morph is promoted. That also was very interesting, and this has been also reported by the people working in the in Max Planck, that they also could find this, and they are working upon, and even I collaborated with them. So here this actually this is a wordy, but what I wanted to say here, that the variations, if they reduce fitness in any type of species, then you will find that after that the selection, natural selection, that favors that type of mechanism that makes this phenotype less sensitive to the environment changes, if any type of adverse effect is there. So in that case, here you can find that the groups, when we try to work on, so this is these are the two different types of morphs that you can see, and in the group we could also find that there were different groups of nematodes which showed this polyphenism or diphenism in the nematodes. But again, there were others where you will find that these two types of stomal arrangement is not there. That means these were having an edge over the others. Pristioncus, that is again another model, Pristioncus pacificus, that is showing this type of diphenism. And compared to that, we also could find one species in our sample where not only the two types, but there were three types of individuals we could notice. And here you will find that the normal type of individual, which was having the teeth they were just smaller teeth, but then we could find that there were other individuals which were having the stenostomous condition with very large hook-shaped teeth. This was the same species, and they were actually the individuals within females. There were two types of females we could find. You can find that the teeth most of the time that was just protruding out of the stoma. And in the same group, this is actually the genus Fictor. Fictor platypapillata, that was a new species, and when we went for molecular characterization and compared with other species, that was markedly different from other species of Fictor. And in Fictor, no such type of biphenism is reported. They are having similar type, but this species is having an advantage where you can find the hook-shaped oncia or hook-shaped teeth, they are present. So particularly we found that this is actually a further step towards predation, where the fast moving individuals are also hooked. And then, so if there are two types of prey, one sluggish and another fast moving, so sluggish one can be attacked, but the fast moving one are also entangled using these, and then they can be. So this is actually again an interesting thing, and we are still looking for others. Likewise, there was another study which was reported, where you can find that the same in the culture, there were two sets of nematodes, that is Acrostichus, which is a bacteriovore, and then there was Pristioncus mayeri. So what happens that whenever, like in the initial stage, you will find that when there is no competition, so Pristioncus will be having the stenostomus type of, but when the other species is there, then they are switching over to the Eurystomus type, like wider stoma, and they start predating on the other species just to eliminate it from the population.
Yeah.
Okay, so this is actually, I think, yeah, so this phenotypic plasticity which has been observed, that also is reported in some groups of nematodes, which is basically an indication how the species that changes in that that evolves in the environment when there is competition. So you will find that this is an example of character displacement, where any type of morphological character that changes gradually with time, and then that makes it more compatible in the environment when it is in competition with species. Likewise, we also could find a similar type of result where we could find 4 type of morphs in the nematodes which were parasitized, which were present in the fig, because they actually were found from the wasp, fig wasp. And there were earlier 5 morphs found, but we could find 4 morphs in a species of fig which was reported from India. There also you find that these are showing different types of gills. That means there are the ones which are bacteriophagous, and there are ones which are the predators. That's all.
Thank you, Professor Qudsia, and I open this for discussion. Only one? Can you give?
I have a question. So if you know that gut microbiota has the metabolites, and they modulate our so many physiological functions, and nematodes also stay in the gut. So do they have any crosstalk, like a presence of gut microbiota derived metabolites? Do they somehow modulate nematode growth or survival?
In the animal parasites you are talking about, yes.
Even in the human gut, I think human gut there are nematodes.
I know.
Human gut there are gut microbiota derived metabolites which regulates our so many physiological functions. Now we know.
Yes.
So what are their synergy or crosstalk between these nematodes in the human gut and the gut microbiota metabolites?
Yes, yes. There could have been, because you have just identified a very, you can say genuine type point, that because these interactions can always be there. In the environment they are there. You can find that both are interacting. Sometimes the nematodes are affected, sometimes they are not affected. Like even in the rhizosphere, which when we studied, we could find that presence of certain bacteria that affected the presence of the nematodes. A certain species of nematodes were found, but not other. That's why the rhizosphere of the plants, they actually indicated very different nematode community. If you see one plant species and then another plant species, you will find that the nematode community is different, because the associated bacteria and the fungi, they also are affecting the population of these nematodes. So there is definitely an interaction. But I'm not working on that, but definitely, as you say, there is an interaction, because they are, because both of them have to survive. So this is basically a type of compatibility that they have to establish. Yes. Of course, sometimes the bacteria they overcome, they can be pathogenic and they can kill. So they can eliminate the population. This also happens. Some of the pseudomonas which are there in the roots, they actually eliminate the population of the plant parasites. So this also happens. It is basically the soil frontier, it is very complex. You cannot predict one condition. There are multitude of factors which may affect. So if I do something in the lab using one factor, there may be other factors which may also come into play. Yes.
So I have one question. Ma'am, what are the impacts of the synthetic pesticides on these nematodes, which are basically a biopesticide?
Biopesticides.
Yeah, these are biopesticides, and you know the usage has gone up nowadays.
Yes,
The
yes.
pesticide, and there's decline in the population of the nematodes.
So particularly you are talking about EPN, the entomopathogenic
Entomopathogenic
nematodes.
nematodes.
So basically they because they are always sprayed on the surface of the plant. So one thing is that they always require a type of ambience which may not be very harsh for them, because nematodes are basically aquatic organisms. They always require a film of water to move. So if the surface is completely dried without any film of water, their movement will be hindered. And then on the top of that, if you are using the pesticides, basically they also are detrimental. That's why this is another option of using these EPNs for controlling the pest. Though you can say that is they have got a lot of success, because now they have commercialized it and they are now used in powdered form, because they are up to a great extent they are showing anhydrobiosis. Yeah.
Thank you.
So thank you, Professor. Last question.
Yes. There are so many different kind of laws. So there could be two processes. One is that these are reactions to environmental cues.
Yes.
Another could be because there are so many species which are sympatric.
Yes.
So as a result of niche partitioning,
Yes.
so they develop these different traits.
Yes.
Now when if it is in sympatric species, this kind of development of these traits, not so much in allopatric species, does it also lead to resource partitioning, for example?
Yes, yes, that is there, because you see that they are there in the fig, okay? So they appear at different stages also. That means they are not at the same time, they are not present. You will find at different stages you will see. So those which are bacteriivores, they are initially, they are there. After that, there is dominance of the predators. And then, so there is, you can say condition of resource partitioning as well. In sympatric species, yes, yes. Because they are sharing the same environment, yes. So there is.
Thank you, madam. So I thank both the speakers, and it's a very good and interactive session. And I hand over.
Okay. Thank you. Thank you so much, sir, for chairing this session. And we are not going to leave you, because he is our next speaker. May I now request Professor Raghavendra to chair this session. Professor Raghavendra, please.